All right. In the next segment, I want to talk more about the function of the different types of organelles within cells and their function. All right. And as you can see here, I have bulleted out the major internal parts of a cell that I want to discuss. Now, the first one, the systole. Think of remember we mentioned cytoplasm and when we started talking about the three major parts of a cell. We said that cells have a plasma membrane. We said that cells have organelles in the cytoplasm, and then we said that the nucleus is a big important entity within the cell as well. All right, and what the systole is, think of the systole as basically the water and the non-living components that you find within the cell. Okay, so basically, if you take a look inside the cell, you know that there are all these working parts. There may be a nucleus, some ER, Golgi, ribosomes. Okay, and then there's a lot of water within the cell as well. Okay, that would be the systole of the cell. All right, and then any proteins or molecules or ions or whatever that would be found within here as well. Okay, so the fluid within the cell and the non-living components that are dissolved within the fluid make up the systole. All right, and then the nucleus of the cell, that is basically the control center of the cell. I'm going to talk more in depth about these individually in a little bit. All right, and then you've got what are called the endoplasmic reticulum, the rough endoplasmic reticulum, and the smooth endoplasmic reticulum. Now, the endoplasmic reticulums are often abbreviated ER, okay? And when we're talking about the rough endoplasmic reticulum, we would abbreviate that as the RER, and the smooth endoplasmic reticulum would be the SER. Okay, so keep that in mind as we're going through our discussion, and I don't feel the need to write all these out when we can just abbreviate these terms. All right, and then we've got something called a Golgi complex, or it's often called the Golgi apparatus as well. Either way, I don't care how you use it, just as long as you use it in the correct context. And I typically call it the Golgi complex. So and we'll talk more about the function of the Golgi complex. And ribosomes. Ribosomes are very important little structures within the cell. These are basically the sites for protein synthesis. We've already started talking about mitochondria a little bit, and we said that mitochondria within the cell is where the bulk of ATP is generated. Remember the fuel so the cell can perform the bulk of its work. And then cells also contain these little structures, these little pouches called lysosomes and peroxisomes that are basically used to, think of it as keeping the inner workings of the cell clean. I'll talk a little more about those again in a bit. And then cells also have a skeleton as well, have a framework that give them a specific shape. All right, so on that note, let's kind of move on. And so we'll start by taking a look at this general representation of a cell and talk about the individual parts that we see here within the cell. So as you take a look at this image, what you'll notice, there are various parts within here. Now, bear in mind, as I keep mentioning, that these cells are found in very watery environments, okay? The systole of the cell, there's a lot of water inside the cell, the extracellular fluid, lots of water around the outside of the cell. Okay, so as a result, this water is constantly shifting and moving around, and as a result, these organelles are always constantly shifting and moving around as well. The nucleus isn't always smack dab in the center of the cell, okay, like you always see it drawn in the books. The nucleus shifts around a little bit. This ER is going to shift around a little bit. Okay, so bear in mind that these cells are very dynamic structures, because remember the fluid mosaic model that we started to mention when we were talking about the plasma membrane, the fluidity of the cell membrane into the cell itself. Okay, the cells are very flexible yet tough, durable little structures. All right, so now as we take a look at this cell here, we can see a few different parts. So first, let's talk a little bit about the cytoskeleton. Okay, the cytoskeleton. Okay, or the framework of the cell. All right, so as you take a look here, if you look inside the cytoplasm here, you can see what looks like a bunch of filaments. Okay, see all these stringy looking objects within the cell. Okay, those filaments are structural proteins that form the framework of the cell. Because in the next chapter, when we start covering histology, one of the very important concepts you have to learn about histology when it comes to understanding what tissues look like. If you want to identify tissues, you identify tissues by looking at the shape of the cells that are within the tissues. And there's other things you look at as well, but that's one of the big ones. Okay, and depending on the proteins and the arrangements of these proteins within the cytoskeleton will give cells certain frameworks and shapes as well. All right, and there are various proteins. There are microtubules. Okay, there's actin. Sometimes you'll see some myosin. Okay, and other proteins that that form a framework within the cell. And then what these proteins tend to do is they tend to form kind of a scaffolding-like network around each other. And like I said, that as you arrange these in a specific fashion, that will give the cell its shape. And realistically, that's all the that's all the cytoskeleton does. It's like our skeleton; it just gives us a framework and a shape. Now you'll notice here, as we kind of take a look within the cell as well, that there is this spindle-like structure found in here as well. Now that's comes as a result of the cytoskeleton as well. This spindle apparatus, or what we're called the centriole. This is very important for the next topic I'm going to be covering, mitosis. Okay, cellular nuclear division. So nuclear division, cellular reproduction, however you want to call it. Okay, because these spindle fibers here, you'll notice that as you take a look at this spindle structure here, there's a lot of proteins projecting off of there. All right, and what happens basically when we want to divide a cell, what happens are these spindles migrate to each pole of the cell, and then what they do is they pull on chromosomes of the genetic material, and these spindles kind of branch out, grab onto the chromosomes, and then they pull them towards each pole. All right, and then the cell's going to split in two and then form two genetically identical daughter cells. All right, so these are very important. We're going to come back to these when we talk about mitosis and nuclear cell division in a little while. All right, but that's all those are, and like I said, those are derivatives of the cytoskeleton as well. All right, so the cytoskeleton, forming framework, and also plays a little bit of a role in mitosis or cell division. That's all you really need to know about the cytoskeleton, framework of the cell. All right. Now, as we kind of continue and take a look at some of these other structures, I mentioned that there are what are called lysosomes and peroxisomes. Okay, as you can see, these kind of sac-like structures here within the cell. All right, what these are, if we, lysosome or a peroxisome. Okay, LYS in medical terminology means, think of that as to break. Okay, think of lyse as to break. Okay, so if we're talking about lysosomes, what these are little pouches or packets that contain enzymes, okay, digestive enzymes. All right, so basically what these lysosomes do then is these enzymes break apart materials, organic materials. So for example, remember we talked a little bit about phagocytosis, not even a little bit, it was very brief, about phagocytosis. We said that there are phagocytes that they wander by these foreign materials and they create these invaginations in their cell membranes. Okay, and then they create these little sacs called phagosomes, and then what happens? Okay, so then let's say this green dot represents a bacteria. Okay, and then what happens? Once this phagocyte phagocytizes or eats this bacteria, then what's going to happen is a lysosome is going to fuse with this. All right, it's going to fuse with this, and then these enzymes that are inside of here are going to are going to be basically dumped out or released into the phagosome, and excuse me, and then that's how this phagocyte is going to digest this bacterium or whatever foreign material that it consumes. All right, so lysosomes are basically used for digestion purposes, and we need them for other things as well. I mean, it's more than just phagocytes utilize those lysosomes. I mean, for example, if we have a mitochondrion or this ER or Golgi or whatever it is we're talking about here, if we have, organelles get worn out. Okay, organelles, like anything else organic, are going to fall apart over time. All right, but we can replace these old parts of the cell, these organelles of the cell, but just like a part in your car, for example, if your alternator goes bad, you have to take the old bad alternator out before you place a new one in there. Okay, so we have to get rid of the bad part, all the bad fragments and the useless part, before we can actually build a new one and replace it. All right, and that's what lysosomes are going to do, is they're going to break down that old worn out organelle as well. All right, so that's all they do, and they also play a role in something called autophagy, or we can think of another term for it as apoptosis. All right, because there are certain situations when cells just get rid of themselves. Cells, a lot of people like to call it cell suicide. Okay, and basically there are certain situations when this does happen. For example, if a cell takes on a lot of damage, for example, if we develop a cancer cell or a bad defective cell, and a natural killer cell or a cytotoxic T cell, cells of your immune system. What they'll do is they'll basically fuse with this bad cell. They'll dump a bunch of digestive enzymes, or called granzymes, inside this cell, and that what'll happen is these enzymes will break this apart from the inside out. Okay, and then what'll happen is, let's say this is a tumor cell. This cell will be so damaged on the inside, it'll be so damaged beyond repair. The only option it'll have is to kill itself. Okay, and then the lysosomes that are within the cell are going to start to break open, release the enzymes all over the place within the cell, and then that will break apart the organelles and basically be the demise of the cell itself. All right, so that's basically what lysosomes are there for. Phagocytes use them to digest whatever it is they consume. We use them to replace worn out organelles, and they're also involved in apoptosis. And what's interesting, I mentioned the apoptosis as well, is that what's weird about it is there are certain situations where we don't exactly know why it happens. Okay, for some reason, some cells just get rid of themselves, and we don't know exactly why. All right, but but apoptosis is if you go more in depth with it and study it, it's a very interesting phenomenon that occurs within cells. It's a very systematic way that cells have of getting rid of themselves. All right, and peroxisomes do essentially the same thing, except for they use peroxides. Okay, like hydrogen peroxides and so on, to break down more inorganic type materials that could get into cells as well. All right. So that's basically what peroxisomes do. They take care of the inorganic material. Lysosomes take care of the organic material. All right. So basically, in a nutshell, these two are keeping the cell clean. All right. So lysosomes and peroxisomes. All right. So those are kind of the oddballs, the cytoskeleton, lysosomes, and peroxisomes. Now let's talk about the major, really working parts of the cell. All right. So there's a few different parts on here. First, we have a nucleus. Then we've got some mitochondria. Okay, and then you can see there are these kind of invaginated folds here. This one is the RER, the rough endoplasmic reticulum. This here is the smooth endoplasmic reticulum. All right, and then this is the Golgi complex. All right, and then we mentioned, the spindles and the fibers, the parts of the cytoskeleton. Now, what these are, these little dots you see all over the place out in the cytoplasm, these are ribosomes. Okay, those are ribosomes. And then we've got some It could be peroxisomes or lysosomes. All right. And then everything else you see is the systole, the fluid, and the non-living components that all these working parts are kind of bathing in within the cell. All right. So let's talk about all these various parts of the cell and how they work. Now, what's fascinating and interesting about these different organelles is that they're almost like an assembly line. Okay. So basically, you know, we've been saying this whole time that cells are the functional units of life, okay, the smallest working parts that can give us life, you know, because when I mentioned my hierarchy of complexion, you know, I said a mitochondrion alone is not good enough to give to sustain life. A nucleus is not good enough alone to give us life, and ER and so on. But when we take all these individual parts and we put them together, then we can form a living entity, i.e. the cell. All right, and then these diverse cells then give us function and give us life. All right, and like I said, you know, as I mentioned before, you know, keep in mind as I'm talking about this that no cell in the body is going to look exactly like this, okay? Because cells are going to have, I mean, almost all cells are going to have a nucleus, but they're going to have varying amounts of this endoplasm, of this rough endoplasmic reticulum, of the Golgi, you know, mitochondria and so on. Okay, so that's where you see the diversity in the function of cells. All right, is within, you know, when you look at these under a microscope or images of cells under a scanning electron microscope, and you see how many different, you know, the variety of different parts and how many of the parts there are. That will dictate a lot of the function of the cell. So understanding these organelles can tell you a lot about the jobs that these cells play. So as you're going through and learning about A&P, and you are, you know, studying the cellular composition of certain tissues, you know, understanding the basic functions of these organelles are going to tell you a lot about the jobs that those tissues play. All right. So on that note, let's start talking about the different these organelles and how they work. All right. First, let's put our focus on the nucleus. Okay, let's put our focus on the nucleus. Erase all that. Just do this the easy way. I always miss something. All right. So first we'll talk about the nucleus. Okay, the nucleus is the largest organelle that you find within the cell. All right, and the nucleus is, you know, not only the largest organelle, it's probably the most important organelle as well. All right, now within the nucleus, you know, you're going to find DNA. Okay, that's where we find the DNA of a cell, and, you know, as we're going to talk about later on, DNA is basically what makes us who we are. Okay, DNA is what we use to synthesize proteins. It's a code. All right, so within the nucleus itself is where you find that. Now, it's important that we store DNA inside the nucleus because DNA is a fragile molecule, all right? And damage to DNA could cause, you know, could lead to or cause mutations in a cell, and that's really going to, that could vary, well, mutations almost always negatively affect cells. Okay, and I mean, for example, if we damage DNA enough in certain cells, that could cause, you know, them to make bad copies of themselves and form into cancer cells, all right, and so on. Now you'll notice when you take a look at this DNA that it has its own little parts to it as well. If you look at this dark inner portion, that's the nucleolus. Okay, that's the nucleolus of the nucleus, and then you can see that it has an inner and an outer membrane, all right, and this outer membrane is what we call the nuclear envelope. Okay, it's the nuclear envelope. All right, and then what you'll notice, what you'll notice next are that there are these pores, okay, that we call nuclear pores. All right, those nuclear pores are very important as well. All right, so the nucleus has a couple different parts to it, as I just mentioned, and these parts do serve various purposes. So basically, So basically what we're looking at here are when we look at these different parts. Now, one thing you need to keep in mind about this is that DNA does not escape the nucleus. Okay, DNA does not escape the nucleus. All right, but all these various almost all these parts out here in the cell are used for synthesizing and making proteins. All right, and now I just mentioned earlier that DNA is the code for making proteins. So obviously, if DNA cannot leave the cell, there has to be some other way of doing this. All right, and basically we use what's called RNA, more specifically messenger RNA. You know, it's a message template that's formed from DNA, and that RNA travels out these nuclear pores to the protein making machinery of the cell. All right. Now you have to bear in mind that's all cells do. That's the number one job and function of a cell, is to crank out and synthesize various proteins, because these various proteins are what give us life. You know, the proteins that make up the structural composition of the cartilage in your knee, the proteins that act as enzymes that perform chemical reactions within the cell, proteins that can be used within the plasma membrane as receptors or gates, or proteins that code for eye color. Okay, all those proteins that drive life are synthesized from DNA. Okay, and all of this here you see within the cell, that's where you find all the chromatin, or kind of the tightly packed stored DNA within the nucleus. And then this nucleolus, this very, very dark area, that's basically where you find DNA that's used to make what's called rRNA, or something that's very important in producing ribosomes and making proteins. All right. So that's basically what we use the nucleus for. We store DNA in there. All right, we store the message to make protein. All right, that message then is transferred out of the nucleus through these pores out to the protein making machinery of the cell. All right, I'm going to talk much more about this in depth later on when I talk about transcription and translation, and how we, and cellular division and so on. But that's really all the nucleus is, and that's why we call it the command center of the cell, because that's where DNA is stored, and DNA basically directs all the activity of the cell, all the protein making activity of the cell. Because that's, when you look at this image, that's the bulk of the job of a cell, making proteins. All right. So then next, when we take a look at this, let's take a look at the rough endoplasmic reticulum next. Okay. And the reason why we call it rough is because you'll notice that there are that it looks very granular in appearance. Okay, and if you look at this under an electron microscope, it is going to look very rough in appearance. And what these little specks are, those are ribosomes. Okay, and as I mentioned before, ribosomes are the sites of protein synthesis. So you can guess then what the rough endoplasmic reticulum's main job is. Okay, this is where we make proteins. Okay, more specifically, proteins that are to be exported. Okay, so typically proteins that are synthesized in the rough endoplasmic reticulum are exported out of the cell. All right, so now if we want to look at the design of the endoplasmic reticulum, what you're looking at here is an extension of the nuclear envelope. Okay, so the rough ER is essentially just, it's an outpouching or outgrowth of the nuclear envelope into the cell. And then you'll notice that the rough endoplasmic reticulum has a lot of folds. It's a very folded type organ, all right? And basically as it folds like this basically allows us to conserve and save space, all right? So you notice we can store a lot of ER within the cell. All right, so that's all you really need to think about this. So for example, let's apply this rough endoplasmic reticulum. Let's say you're talking about a B cell, all right? B cells are cells that we use to produce antibodies. Okay, you guys know that antibodies are proteins that we use to combat infections and illnesses. All right, so what happens, so let's say you've got a viral infection, you've got a flu. All right, if this B cell comes in contact with this virus, then what it's going to do is it's going to start to divide, but it's going to form these very big looking cells called plasma cells. All right, And these plasma cells, all they are, they're basically just nucleus and almost all rough endoplasmic reticulum. Okay, they're almost all nucleus and rough endoplasmic reticulum. All right, and this rough endoplasmic reticulum, like I said, its main job is for producing proteins that are to be exported out of the cell. All right, so this is where these antibodies come from. That's the main job of this. So then this plasma cell is just going to sit there and synthesize antibodies at a very rapid rate that are going to be excreted into your body fluids, so you can help fight the infection that's infecting you. Sorry, I can't think of a better way to put that. Okay, so that's the rough endoplasmic reticulum. All right, so the DNA in the nucleus then is shuttled out to these ribosomes on the rough ER, and these proteins. Then what's going to happen as we start to synthesize these proteins within the rough endoplasmic reticulum? They're going to be enveloped within these sacs. All right, they're going to be enveloped within these sacs, and eventually what's going to happen is they're going to come in contact with this next structure called the Golgi complex. Okay, the Golgi complex. Okay, so just like the rough endoplasmic reticulum, all right, proteins that pass through the Golgi complex are going to be exported from the cell. All right, now the Golgi complex, you'll notice, it looks very similar to the rough ER, except for the fact that it doesn't look granular in appearance. Okay, there are no ribosomes on it. So the Golgi complex is not responsible for making proteins, but it can modify proteins. This is where proteins become conjugated in cells, where we'll tack on a carbohydrate or a lipid. Okay, this is where a lot of carbohydrates in general are synthesized. Remember we mentioned that the plasma membrane is made out of phospholipids. All right, a lot of the phospholipids are designed here as well. So basically what's going to happen then, as we develop these proteins in the rough ER, and then they'll migrate and pass through the Golgi complex, then what will happen is they'll be in these vesicles, these vesicles. And remember we talked about exocytosis in the previous lecture, and these vesicles then are going to fuse with the plasma membrane, as you can see going on here. And then once they fuse with the plasma membrane, then a couple of things may happen. If this is a bunch of protein to be, like a neurotransmitter or a hormone or a chemical message of sorts, then they're just going to be released out of the cell itself. Sometimes these proteins or molecules, like the phospholipids, are going to fuse with the cell membrane and then become a part of the plasma membrane. Okay, but they're still being exported out of the cell, but they're on the outside of the cell and serving a function to the cell itself. All right, so you notice that we have this assembly line here that we use to synthesize proteins, because like I said, that's the main job of the cell, putting together proteins to either be exported or to be used by the cell itself. This eraser is not working. All right, and as I mentioned before in the previous talk, when I was talking about osmosis and tonicity, what the heck? Okay, when I was talking about osmosis and tonicity, all right, remember that these parts of the cell are, in order for all of this to happen, they need to be a certain distance from one another. All right, so if we become hypertonic and the cells and basically water escapes the cell via osmosis, all these parts are going to get scrunched together and it's not going to work. All right, it's not going to work properly. And vice versa, if our body fluids become hypotonic and the cells, if water goes into the cell and the cell swells up, okay, all these working parts are now going to be pushed farther away from one another. And again, like an assembly line in a factory, it's not going to work properly. There's a reason why when you look at assembly lines in factories, there's a certain distance between where they make all the parts. Okay, because if you're too close together, you're going to just be, you're not going to be able to work properly while you're banging into each other. And if you're too far away, it's going to take too long to make the parts. So the spacing is important as well, and that's why maintaining water balance is important. Okay, so the nucleus is where we store DNA. All right, and remember, DNA is a code to make proteins. All right, so then we use that code, and that code goes out to the rough endoplasmic reticulum, and the ribosomes are the specific sites all right, where proteins are synthesized. And then once we synthesize these proteins, then basically, you know, these with these folds of the ER, they form these sacs, and then the proteins are stored within them, and then they'll migrate to the Golgi. And then, like I said, the Golgi is going to modify the proteins. It could make, like I said, conjugate whatever it may be, and then as these vesicles pass through the Golgi, then they're going to fuse with the cell membrane, and like I said, either be exported or, you know, completely out of the cell, or be exported to the plasma membrane itself. All right, so that's the nucleus, the rough ER, and the Golgi. Okay, now you'll notice that again there are some ribosomes out here, and there are free. We would call those free ribosomes. Okay, and just like the ribosomes attached that are associated with the rough endoplasmic reticulum, those are also sites for protein synthesis. But typically when we synthesize proteins on free ribosomes, these are proteins that are to be used within the cell. Okay, you know, like replacing enzymes that drive certain metabolic functions, or structural proteins like the cytoskeleton and so on. Okay, so that's the difference between the ribosomes that are bound to the rough ER and the free ribosomes of the cell. Okay, proteins synthesized in free ribosomes are used within the cell. Proteins synthesized in the rough ER are exported from the cell. All right. So next, let's talk a little bit about the mitochondria. All right. Now the mitochondria are very important because that's where, as I mentioned before, that's where the energy comes from to drive all of this activity within the cell. All right. And basically, remember we've already started talking a little bit about ATP. I'm going to talk more about ATP when I talk about metabolism later on. Okay, but basically, this is where the bulk of ATP is synthesized. We can make ATP within the systole of the cell through a process called anaerobic respiration. All right, but that's not we don't make a lot of ATP as a result of that. It's not a very effective mechanism to make ATP. All right, so basically most of the ATP is produced within the mitochondrion through a process called aerobic respiration. When we say aerobic, okay, you should basically think we're using oxygen. Okay, we're burning oxygen. If I say anaerobic, okay, I'm saying without oxygen. All right, and then, like I said, as you'll learn as I talk more specifically about the steps of this in another lecture, that making ATP with the use of oxygen is a lot more efficient, and we yield a lot more ATP as a result. Okay. So, remember before when I was talking about proteins in chapter 2, the average protein is about 25,000 to 50,000 amino acids in composition. Okay, that's all. I mean, that's a lot of molecules. Proteins are big, bulky molecules. They're much larger than carbs and fats and water and many other ions and solutes and particles that you find within the body itself. All right, so we're making big molecules. That takes a lot of energy to make these big molecules. All right, hence the need for these mitochondria. All right, I mean that's mainly what they're fueling is protein synthesis. All right. And there are certain cells of the body that have more mitochondria. Typically this is easy to think about. The more active the cell, the more metabolically active the cell, the more mitochondria are typically going to be within that cell. So when you start to think about, well, what are the more active tissues of the body? Well, an easy one that pops into mind is muscle tissue. Okay, muscle tissue is very metabolically active. We're constantly this is a constantly active tissue. You burn a lot of calories at rest, via muscle tissue. So therefore, and plus, to sustain something like exercise and movement, that requires a lot of energy. All right. So as a result, you're going to need to produce a lot of ATP so you can perform work. All right. So muscle tissue, muscle cells have a lot of mitochondria. Neurons tend to have a lot of mitochondria. They're very active cells as well. I mean, the brain, you know, in terms of total surface area of the body, is one of the, or I should say, size of organ, is one of the smaller organs of the body itself. But it's one of the most demanding organs metabolically of the body as well. All right, so there's a lot of mitochondria in neurons. All right, to fuel the activity of the cell. So a lot of books and a lot of people that teach classes like this like to nickname mitochondrium the powerhouse of the cell. Okay, the powerhouse of the cell, the energy factory of the cell. Okay, and mitochondria are unique because they actually have their own DNA. Okay, when you look inside these folds called cristae of the mitochondria, there is actually DNA in here. It's called mitochondrial DNA. All right, and what's interesting about this is they, you know, if you look at this from a evolution standpoint, you know, it's believed that mitochondria evolved from other organisms like some kind of bacteria. There's a theory called the endosymbiont theory that describes kind of the beginning of where these complex animal cells came from. You know, from the fusion of a mitochondrion and then other parts to make a cell. So, and I'm not going to talk about that because that's not really relevant to this topic, but it's just something interesting. No, look it up, the endosymbiont theory. Okay. Endosymbiont theory, I'll write that down for you. Okay. So that's what mitochondria do for you. All right. So if you put this all together, all right, you're starting to kind of get a picture here. You've got a control center that's used that's used as a code to make proteins. All right. And then you have all of this machinery around here. You have all of this machinery that's used to synthesize proteins, to package proteins, to create proteins to be used by the cell. That's the bulk of the job of a cell, making proteins. That's life, folks. I mean, synthesizing diverse proteins. All right. And then the last organelle that I haven't talked about is the smooth endoplasmic reticulum. See if I can get this to work for once. Close enough. Okay, so the smooth endoplasmic reticulum, which you see right here. Okay, so basically the smooth endoplasmic reticulum is an extension of the rough ER. All right, the rough endoplasmic reticulum, but obviously we call it smooth because it looks smooth in appearance. It looks agranular in appearance, all right? And the smooth endoplasmic reticulum has quite a diversity of functions. I mean, and it depends on, you know, what types of cells you're talking about. Okay, so for example, liver cells have a lot of smooth ER. Okay, hepatocytes, all right, because basically within the liver is where we tend to deactivate steroid hormones and drugs, all right. So basically what happens are these drugs, they get into, you know, they get circulated in the liver eventually, and then there are, because you got to remember these organelles, they're not like a part in a car. These are packets these are complex packets that contain enzymes that carry out chemical reactions, all right? So the chemical reactions that occur in the smooth ER are to basically take care of or deactivate or denature or render these non-functional. Okay, so for example, let's say you drink, let's say you drink beer. Okay, you drink some alcohol. All right, you drink some alcohol. And what happens then is that alcohol is going to get into the liver, and then the enzymes within the smooth endoplasmic reticulum are going to break down the alcohol within the beer. All right, because you have to remember alcohol is an organic poison to us. All right, it just makes you feel good and do fun things if you have enough of it. So now, basically what happens, now your liver is going to adapt to this, because remember, the nucleus is a site for synthesizing proteins, not just to be exported, but to be used by the cell itself. All right, so let's say, let's say you're talking about building up a tolerance. Okay, you build up a tolerance. So you hear about, party animals in college talking about this, or alcoholics. Okay, so what happens then is as you consume more alcohol, what happens is you develop more smooth endoplasmic reticulum within the cell. All right, and then as you develop more of this smooth endoplasmic reticulum, you're more acclimated to breaking down alcohol. You're much better at it, and that's why a person has to drink more to, you know, get whatever you're trying to get out of the booze. All right, and if we need to make that adaptation, the DNA within the nucleus is going to allow that to happen, because all we have to do is we make the right code to make the right proteins, and then we, create more smooth endoplasmic reticulum, more enzymes within that ER, and then we can handle that. All right. So, I mean, that's what the smooth endoplasmic reticulum does in the liver. All right. In muscle, specifically skeletal, I'll use skeletal muscle as an example. All right. The smooth endoplasmic reticulum in skeletal muscle is an area for calcium storage. All right. And an area was called the SR, the sarcoplasmic reticulum. All right. So basically, what we do is we store this calcium within smooth muscle. All right, within muscle, and then when we need the muscle to contract, the calcium is released, and then we'll carry out the right physiologic mechanisms to undergo a muscle contraction. All right, so like I said, the function of smooth ER depends on what cell you find it in, and these are just a couple of examples. But for the most part, it's used for storage or breaking down other particles or molecules that aren't really useful to us, so that we don't need anymore. All right, so hopefully this kind of helps to paint a better picture of the cell itself and the main working parts of the cell and the overall job of the cell. Like, as I mentioned, the cell is the primary purpose of the cell is for making proteins. The nucleus is where the DNA is stored. The rough ER, making the proteins be packaged and excreted, or the ribosomes that are used to synthesize proteins for the cell itself. Mitochondria and fuel, generate enough ATP to fuel this process, because as I mentioned earlier, the proteins that are 25 to 50,000 amino acids in size, the average size of a protein, all right, we have to expend one ATP molecule for every amino acid we synthesize. So that's a very energy taxing process to make a protein. So therefore, we need an efficient organelle like mitochondria to generate a lot of ATP for this process. Okay, so again, if you have any questions about this, let me know. And I strongly encourage, as you watch this video as well, that you have your book open and some pictures of these parts to take a look at them as I'm explaining them as well.